Lovely. Thank you very much. So thank you very much, Tom, for the introduction to and the invitation. I'm from UNSW Sydney in Australia, and my thing is looking at soft matter at interfaces, particular polymer surfactants, but also looking at things in confinement. And so I just want to dip through some recent work that we've done in confinement and talk about the reproducibility or otherwise of that work. And for those of you who are looking for some slides to download and scribble on and whatever else like that, the URL there at the bottom, deb.li/also2021, has the slides for you. Feel free to pop them on the reflectometry.org website as well. So today I want to talk about a few different aspects of reproducible and reproducibility in neutron reflectometry. So the first thing, I guess, is to define the problem space that we're working in. What do we actually mean by reproducibility? What do we mean by reproducibility in the specific context of neutron reflection? And why do we care about these particular aspects of doing our experiments? It's a good chance for us to think a little bit about what has already been achieved, to look at some of the good practice that is out there in the literature for reproducible and reproducible neutron reflection experiments, but also to look at a little bit about what some of the friction is in trying to achieve reproducible reporting of our experiments, and think about what blockers this group might be able to address as next steps towards making sure that we have nice reproducible stories to tell in our experiments or from our experiments. So I guess the starting point we should start with is, what do we even mean by reproducibility in the context of doing scientific research and publishing scientific outputs? So this isn't just whether you can go and do a similar measurement on the same sample and get the same numbers. This is a much deeper concept than that fairly base level of whether your sample and measurement is reproducible. It's about the ability to figure out, based on reading the paper, what anybody has actually done. And so it's not about whether me as an author has the ability to figure out what's going on. It's about whether you as a reader of my papers have a chance of figuring out what's going on. So this is about, how does the data get collected? This is about how the data is transformed, processed, how it is analysed, and then how do we get from analysis to logical conclusions and outcomes. So public availability of data at various steps in the pipeline of analysis is really important, and public availability of the processing methods is really important. So the National Science Foundation in the US has broadly defined reproducibility as in terms of whether you can make use of the data, whether somebody else can make use of the data, and whether somebody else can make use of the analysis methods. And their comment is that reproducibility is the minimum necessary condition for a finding to be believable and informative. Basically setting the bar that if your work is not reproducible, then it's probably unbelievable and uninformative. And as scientists, I think we would rather that our papers were considered to be believable and informative. So this is why we're starting to think about what we really mean by open science, reproducible science. There's lots of different terms in the stable of reproducibility that we could add in here. Now, I will comment that the biologists and the medical scientists tend to be a long way ahead of most physical sciences in terms of thinking very carefully about reproducibility. And those of you who are in maybe working in biology, biophysics, etc., will already be looking at this and rolling your eyes, saying you guys are only just discovering this. Those of you who are instrument scientists who have been working with people from biology and biophysics will have been subjected to some of this in the past, and where they're saying, well, what did you do to my data? And the answer is black box, and they go, not acceptable. So I guess what we're starting to realize is that, yes, medical science and a lot of biology is a long way ahead of us in being able to talk about reproducibility and being able to talk about what's required and what is desirable. Now, part of that is, of course, because some of the biology and biological sciences and medical sciences have had lots of problems with bad actors within the scientific community, which means that reproducibility is part of a way of ensuring that the particular microscopy image you are looking at has not been photoshopped. etc. Now, we might look at things like retraction watch, and we understand that there are bad actors out there who are doing horrible things to data and who are making their data up, etc. But reproducibility goes a lot deeper than just saying this is about trying to make sure that everybody is being a good scientist. This is about helping each other. And in fact, I would like to convince you today that it's also about us helping ourselves to do good research as well. So let's just talk through a neutron reflection experiment and think about some of the aspects of this neutron reflection experiment where we might consider there to be places where we would like to ensure reproducibility. So fundamentally, to start with, we have a neutron reflection instrument, and this instrument has been floating about in the wild for a while. It's usually a fairly well documented instrument, right? So there's usually a paper that has been written as part of it. Sorry, my screen has just gone nuts here. Let me try and get that back to full screen. So there's normally been a paper written about the instrument. We have normally a nice description of how the instrument works, what it does, etc. But that paper would normally tell us that there are several hundred motors, each of which might do something, and some of which might be very important to how the experiment is done. So the user, when they come to the facility, generally can describe their samples in quite good detail and does so in the experiment, in the experimental write-up. But they're very reliant upon an instrument scientist to help them turn a complicated piece of machinery, the instrument, into a useful measuring device for their particular experiment. And that's why we make instrument scientists co-authors on papers, because they've got a very important role in that. So the instrument gets set up, it collects some data. Almost all facilities stick that data immediately into some sort of raw data archive. Different facilities make that archive publicly available after a certain period of time, etc., for various types of experiments. But that is generally the raw data archive is an indelible record of the experiments that were done, as long as there's enough metadata associated with those raw data files that anybody else, or even the scientists themselves who did the original experiment, has any chance of figuring out what went on the day. Now, we then have this process called data reduction, and that's normally performed at the facility, usually under strong supervision by the instrument scientist, making sure that the correct parameters go into the reduction algorithm. And these might be parameters such as the wavelength range for a time of flight instrument. What wavelength range is going into the reducer? It might be things such as where is the beam center actually located, sub-pixel refinement of the beam center, for instance, on a multi-detector instrument. It might be things like background subtraction parameters. How do you actually figure out where the background is, somewhere around the slits and the sample and the specular reflection, etc., etc. So there is a set of reduction parameters that go into that analysis. Often that reduction is performed in some sort of interactive fashion where either an experienced user or the instrument scientist is tweaking those reduction parameters to try to create reduced data of the highest quality, removing experimental artifact, background noise, etc., as appropriate. Now, you can see that there's a set of questions that are being raised as we progress through this pipeline, such as whether the experiment can be repeated, how the instrument was actually used, how the data reduction was performed. And then we get, once we've actually got some data, well, the next question is going to be, is that reduced data actually available anywhere? The answer, sadly, quite often is no. Once we get to the analysis, that is often performed by the user back at their own institution. There's obviously guidance from the instrument scientist in driving various software. Is the interpretation believable, etc.? And there's a lot of variation in how much of that is done by the instrument scientist or by the user, depending upon the users and their relationship with the instrument scientist. That analysis often has a huge amount of input from a human brain. So yes, there is an optimization algorithm, and there is a model which is in some sort of code, but then the person has often done quite a lot of work in maybe an iterative set of analyses or a bootstrapped style analysis where you're starting from some information that you can measure by other means or by a simpler neutron reflection experiment, X-ray reflection experiment, and then you slowly build up the complexity of analysis. So what actually goes on in that analysis step is often a fairly scary black box, and often the details are poorly reported. So we'll unpack those statements a little bit in a minute. The artifacts coming out of the analysis is some fitted data and some fit parameters, some of which end up getting published and inside the final publication. So that model is maybe reusable or maybe not reusable. It's maybe not disclosed in enough details that anybody can really figure out what goes on. And then the fit parameters might be just summarized to the 2 or 3 most important fit parameters that come out of the modeling in the paper, and maybe the rest of the fit parameters are consigned to the dustbin of history at that stage. So that's our overall reproducible or our overall neutron reflection pipeline. And you can see the questions that I'm asking there raise queries about different steps in this pipeline. And there are 2 levels for these queries. One is, what is even possible for us to describe today? One is, what is in practice actually described today? So what's not just what is feasible to include in a paper, but what is actually being included in a typical paper. So for us to start thinking more about some reproducibility aspects of reflectometry, I want to talk just a little bit about some of the motivations for why we would even want to do this. Now, one of the most simple aspects of wanting to have a more transparent and therefore more reproducible workflow for the reduction and also for the analysis is to prevent things that are just simple accidents. So every single neutron reflection experiment tends to generate lots of files with fairly cryptic 6 or 8-digit file numbers, and some people find those run numbers most edifying and love living in this 8-digit world. Most people, however, sit there and go, all these run numbers doing my head in, I'm going to start renaming files, or I will accidentally get these files mixed up. We all know stories where we have accidentally mixed up run numbers, and we spot them at some stage as we go through the analysis that data doesn't look like this particular this particular sample as it should, or we spot that the background is incorrect, or that the critical edge is in the wrong place for what you think the sample should be, etc. So we can often spot those accidents, but we don't really want to be reliant on being able to spot accidents after the fact. We would like a transparent scheme that prevents us from doing stupid things like accidentally reducing data against the wrong direct beam, for instance. Accidentally getting the background subtraction wrong, for instance. So just in terms of catching mistakes and therefore allowing us to fix them, reproducibility is a huge advantage for us. Now, your users are forever bringing in new PhD students, and the instrument scientists, you are forever training new users on the instruments. And when it comes to onboarding a new PhD student who is a new postdoc, who is new to the reflectometry again, and they say, well, I'm proposing to do this experiment, and I think I'm going to run 500 samples in an hour. And you go, no, you're not. It takes much longer than that. And they say, well, how do I know? Well, how long does each actual exposure take? Does each measurement take? Is that even in the papers? The answer is it often is not in the papers. And then you get to the fundamental details of how do you set the instrument up. Things like what area of the sample is illuminated, what is the footprint, what slits were required to create that footprint. There are some fairly fundamental parameters that we might want to know in a paper to understand what an experiment would look like. And if you know that an experiment is going to take 2 hours on this instrument, and you know that your flux is a quarter of that particular instrument at other facilities, then it's going to be a much, much longer experiment on your own instrument. Maybe you can't do it, maybe it's not feasible. There are many advantages for knowing more about how an experiment was undertaken. Once we move a little bit further into what we might be able to reuse out of our own work, there is the question of model reuse. So we spend a lot of time generating complex models, mathematical models, which describe what is going on at the interface, which is the basis for our reflectometry analysis. Most of our models are actually incredibly reusable because we all study fundamentally similar things. So the ability to pick up a model and make use of it by somebody else or by your next student or by your next user actually helps everybody. So the idea of having reusable models through some sort of marketplace where they are exchanged, whether that be in a paper or whether that be a separate marketplace online or GitHub, for instance, that benefits all of us. Now, the other thing that we have to think about is that we should be able to reuse the raw data. So neutrons are scarce things. We can't just afford to rerun samples just because we've gone from PhD student 1 to PhD student 2 over the course of a long project. And we might want to be able to plot data from one experiment against in comparison to data from a newer experiment, where maybe it's just a different concentration of the same thing. And so you want to be able to make use of that old data to plot a series of reflectometry profiles that show change over time or change over concentration or something like that. It's very common for us to want to make reuse of data. When it comes to writing something as simple as a review paper, we often want to either take hold of old data and plot it in a slightly different way or plot it in comparison with somebody else, or maybe even reanalyze the data to be able to make use of a newer model compared to that the older work did not have access to. Maybe we can get more value out of existing data. So reuse of data is an important thing for us as well. Finally, there is that data integrity idea that once we have a clear data pipeline, we can start talking about the hygiene of handling data. And data integrity does, and the transparency in putting together reproducible pipelines for our data handling also does mean that we can say that data manipulation becomes much harder or maybe even impossible. So data integrity is certainly one aspect of reproducibility, and that is the aspects that many other fields have spent a lot of time thinking about data integrity and reproducibility as a result. For us, there are all these other reasons as well that we definitely want to look at too. Now, I'm going to just talk a little bit about some experiments that we have published. And so I need to give you a little bit of context for why we're asking these questions and what these papers are that we're going to dip through. So we've spent a lot of time looking at what polymers do in small geometries, not just polymers, also lipids and surfactants and other sort of similar systems. What they do in small geometries is to provide new functionality to a surface. And that can be responsive, that can be lubricating, that can be stabilizing from a colloidal perspective. But in all of these cases, the question is, well, what does this polymer at the interface actually look like? The polymer at the interface is influenced, is important not for the polymer being there on its own, sitting in an infinite or a semi-infinite amount of liquid, if we want to think of it in those terms, is not actually interesting or useful like that. It's only interesting once we start bringing something else up to it, to within some small geometry. And it's not microns, it's not even hundreds of nanometers or tens of nanometers. We actually really need to get down to separations that are smaller than the typical length scales of the molecules that we're talking about. So when we start talking about polymers, then we're starting to talk about separations that are down at the few nanometers to few tens of nanometers at most. And that then starts looking like a fairly complicated neutron reflection experiment. And the reason why I've picked this as an example is partly because we've got, I can take a slice of papers over the last 15 years, which go through various different ways we have tried to probe this exact problem and look at where we're doing well and where we can still improve in our analysis. Now, one little thing, just because it's a common question that people often get the wrong end of here for talking about and thinking about this particular sample environment that we're talking about. This is not a pressure cell, and this is not about hydrostatic pressure. A coffee press, a cafetiere, is a much better way of thinking about this experiment. It's free draining. So it's asking about what happens to the structure of the coffee grounds in your cafetiere as you push it and you allow the solvent to escape. So as we start to go to higher confining pressures, and we end up talking about confining pressures, we end up ejecting liquid to the point where we have change in concentration of our confined soft matter. So it's about the confinement and the geometries of the systems, not the actual pressures. Pressure is just a knob that we can twiddle in our control box to change the geometry. That's sort of a detail about the sorts of things that we have tried to measure and the sample environment we are actually measuring it with. So what does data look like from these sorts of experiments? So this is some very recent data from a set of experiments that we did at ANSTO on the platypus reflectometer, where we have a thermoresponsive poly N-isopropyl acrylamide polynipam brush, and we have changed both temperature and confining geometry. Remember that we're describing geometry in terms of a pressure that creates that confinement. And we end up with some very, very complicated data that takes quite a lot of analysis. And to be able to put together that analysis, we end up with a distance distribution function. We have a set of different distance distributions for the confining apparatus, and that's then talking to us about the thermoresponse and how that thermoresponse is modulated, and we end up with hysteresis in the system as well. So that's the sort of analysis that we have been trying to do. Now, in terms of describing the reproducibility or otherwise of our analyses, as I said, we've played around with this sort of confinement experiment for quite a few years now. And so I want to take a bit of a snapshot through sort of about 12 years worth of papers, all of which come from me, so that I can feel free to criticize them, so that I can go through and critique where we are doing well and where we are not doing well. This first one I've called a 2010 paper because it really came from work that we did in sort of 2007, 8, 9, but it finally got published in 2012. So to call that a 2010 paper because it was written to the standards of sort of 2010, so to speak. So the material contained in each of these papers was considered perfectly good at the time. They were following or exceeding standard practice for reflectometry experiments at the time that they were written, time they were published. And they're all vaguely about polymer brushes and confinement, so that we're sort of hitting similar problems or similar issues in each of them. It's not like any of these are in crap journals where you would necessarily expect to see unreproducible science either. These are all published in places which know what they're talking about and hold their authors to high standard. And we, of course, have throughout wanted to hold ourselves to high standards in writing each of these papers, but we'll see that through a 2021 viewpoint, we could do better on all of these work. Right. So this is partly me imposing a more modern eye, and with knowing more myself, and also the entire community is now thinking more about reproducibility of data, and I'm now applying that to these older papers, which in a way is deeply unfair to do because every single paper, every single document is a product of its time. So in a way, it is deeply unfair to do this, but that's one of the reasons why I'm doing it to my own work and not to the work of other people. So if we were to start off with sort of looking at the oldest of those those experiments, those were mostly done on D17 and on INTER as the ILL and ISIS respectively. And the instrument being specified is about the only thing that you can say for the experimental details in that paper. There are scant details about anything else to do with how that experiment was done. And in fact, even though we would normally always put proposal numbers, etc., in the acknowledgements at the least, there's not even proposal numbers in this particular paper, let alone run numbers, let alone any mapping of figure 5 corresponds to this particular sample, which corresponds to runs 1, 2, 3, 4, 5 through to 1, 2, 3, 4, 8, etc. The raw data is not available, even though that was ISIS and ILL, because that's data that predates their open data policies. Those experiments predate their open data policies. So in terms of how we're going for the reproducibility of that 2010 data, we haven't really got a set of experiments at all presented in that paper that we could call reproducible. If we move just so 5, 6, 7 years into the future from that original paper, we can start to think about, we would expect to see some level of improvement. But we don't. That 2015 paper, once again, tells you that it was D17 and Inter was the source of the raw data, but there's nothing else there. And particular for things like the footprints and the slits, these are experiments that are very punishing to the hardware that is running the particular measurements. With apologies to Rob Dalgleish, Christy Canane, Rob Barker, all the people who have helped me do these experiments over the years, they would normally have to go off and recalibrate their slits before I arrived, because these tiny, tiny, tiny slits to get really low angles to get small footprints, very challenging experiments to do. So if anybody else wanted to do these experiments, they should know that difficulty in advance. But we haven't managed to put those details into the paper or into the supplementary information for the paper. So moving forwards to our most recent publication on this similar work, we've got a little bit better on some of these things. I was surprised to realize that I had not even remembered to put in things like measurement times, still, even in 2021 or late 2020 when that paper was written and submitted. So we've got, over the course of 10 years, even having thought carefully about things like reproducibility, we're still missing details as we go along. As we start to think about things like data reduction, we start to talk about, well, what run number was matched to what direct beam for the purposes of doing the data reduction. And we see that over time, where we have the details of how the reduction was done are often not clear, often not published, and that is not something that has really got better with time. The most recent work we have, we are at least getting to the stage where the software is usually published, but the details of the data reduction, so the software is available, but the software itself, the algorithm it uses, the parameters that went into it tend not to ever have been published. And we're moving towards more entirely algorithmic reduction, but we're still not quite there yet. And the paper itself has no evidence that it was purely algorithmic reduction. When it comes to analysis, once again, we have improvement over time, but we've gone from a situation where this very first paper here, the optimizer that was used was Rob Richardson's own little hairball of code. Rob is the only person who has ever seen the code to that optimizer. Love Rob dearly, but that is a long way from being reproducible science. We move forward and we've got the use of Rascal, which was at least open source, but the code for the model wasn't available. And our most recent work, as I'll show you in a sec, ends up with the code for the model, the code for the optimization, all being available. So things are definitely getting better in our analysis. So I've highlighted sort of some of the strengths and weaknesses of what we've been doing. Let's look in a little bit of detail about why I'm saying these things, just for this most recent paper. What does current practice look like? What does good practice look like?
Stuart,
and what do we do as next steps? Yes, Tom.
I was just going to say, you maybe got 5 minutes left.
Yep, sure, perfect. Thanks. So our detailed view of the 2021 paper is saying that we've still got a reasonable amount of thinking to do about how we present information in a way that is digestible into usually the supplementary information, one would assume for this. We need to start getting towards peer review, asking for, where is this information as well. The reduction details doesn't have, the paper is short on reduction details for what was done, the fancy pants details of how to polish that reduction, center finding, beam finding, background subtraction. It is mostly all automatic because it's been worked on very hard by Andy Nelson for that, but we're not really talking about that particularly well in the paper. And this is a big omission from the paper, that we do actually have batch reduction there. It is almost all automated. We do have a clear record of how the reduction was done. We didn't include it anywhere in the paper. So this is one of the things that is a sort of next steps, is to have clear standards for reduction of the data as a simple publication, simple white paper, for instance, about platypus data is reduced in the following way using this published method For example, from Philip Gutfreund, here's how you reduce TOF data, here's how you go through it, here's the tooling that was used, and here are the 3 parameters that you need to know to be able to reproduce it. The analysis for this, I just want to highlight a couple of bits of good practice here that we had in the analysis. We have a short description of the paper, obviously, supplementary information with lots more data, and supplementary information with actual code. And supplementary information, the code is not just buried in the PDF in text format, it's actually also attached to the PDF so that it's a couple of clicks to download it or to extract it from the PDF and to then throw it into your analysis. We have a link to the Zenodo record, which is included in the paper, which has all the reduced data and has more detail, the code for the model, etc. as well. In talking about good practice, I would just like to highlight also some recent work by Andrew McCluskey and team, who published this paper, I think it was earlier this year. It's a lovely piece of work which, in terms of describing the reproducibility. So we have here a link to the Zenodo record. We have a link to a GitHub repository. The GitHub repo has all of the reduced data, all the Python files for the model, all the Jupyter notebooks, and all the instructions on how to run the analysis. So this is another example of excellent practice that does already exist in the literature. One caution I will throw in is about centering around Jupyter as the platform for delivering all these things. Jupyter is definitely better than an unconstrained graphical user interface in terms of generating a repeatable and followable trail of what was done. However, we are seeing some limitations to Jupyter. And one is that as a file format goes, it is neither human readable nor particularly machine friendly, particularly with megabytes of encoded binary images thrown into it. It's a file format that is not going to be a record for the future. It's a working document right now. We wouldn't go and pick a Word document as being our record for historical record. We would pick a PDF, and JSON is a bit the same for that. So we need to be careful about relying upon JSON into the future. There are some knock-on effects about how that JSON file format and Jupyter notebooks end up harming us in the future. So just in terms of what we are doing well, we are doing data analysis and we are reporting data analysis well. The tooling is helping us here for this. This is great. As a community, we need to start asking for this more at peer review stage. That's how we spread the word. That's how we start enforcing this. The data transformations we do, we lose metadata all over the place. The current set of tooling and file formats do not help. And we are very underspecified in how we deal with those data transformations. Finally, the experimental details for the sample are normally great, but I suspect that the user, when writing it up, doesn't know the right things to be including for the experimental details of the instrument. And so peer review needs to be asking for those details more as well. Now, I have aimed to be controversial and opinionated on this, partly to stir discussions into the future, because I know you have more working group discussions on exactly this subject. So I have aimed to be controversial and hopefully spark some discussions about where is the tooling helping and where it is hindering. I want to specifically acknowledge Andrew Nelson and Isaac Gresham. Andrew as our collaborator and instrument scientist on many of the platypus experiments that I've done here. He's also developed the Refinex suite, which we use for both our reduction and our analysis. Isaac Gresham, a PhD student of mine who has recently finished, so Dr. Isaac Gresham at this stage, and he has done a huge amount of work on developing the tooling for us at our research group end. So there are a little bit of time maybe for questions, but also hopefully some more discussion in your working groups. Back to you, Tom.